friends, welcome to my channel. So in the last class we talked about the important disease of groundnut and their management also. So today we will be taking about the important disease of sugarcane and their management also. So sugarcane, this is a crop, or we can say it's a cash crop. Okay, this is a cash crop which is affected by a number of disease or pathogens. So most of the important ones include like red rot, smut, grassy shoot and red stripe. These are the most important diseases. So these are the four important diseases that are considered to be a more important economically. So here first one is the red rot of the sugarcane. It is basically caused by the Coletrotrichum falcatum. This is the scientific name of the pathogen, and the pathogen they survived in the infected host debris and volunteer or ratoon plants. So the plant they can be easily diagnosed with the disease where the leaf can be seen having a red center, and when you split open the canes, you can see that the vascular tissues are also red in a color. So this is the symptom. So these are very easy and distinct way of identifying this disease. So now we see the some management practices. So here we see the include IPM practices, that is integrated pest to disease management, which are cultural practices, use of some resistant varieties and chemical control. And the cultural practices also include uprooting of the infected plants if they are only few plants are destroyed. The crop rotation is very important in the in this management of this particular disease, where we should go for at least 3 years of the crop rotation with a non-host plant. Like similarly, use of digit free sets for planting, it is also very important, because the pathogens it is harbors in a infected host debris. Then we can also use hot water treatment of the sets before planting at the temperature is about 52 degrees Celsius for the 30 minutes. Is one of the most effective control measure for the management of the particular pathogen. Now next is the deep fresh sets at the rate of the 0.25% solution of the alcohol, and also एरेटॉन इज़ ऑल्सो रिकमेंडेड. ठीक, वो किसी भी एक को आप यूज़ कर सकते हो. नाउ रेजिस्टेंट वेराइटीज़ wherever available, we should grow those resistant varieties for having disease free crops. like other chemical control भी है यहाँ पर. this includes set treatment with 0.2% थियोफेनेट methyl, ठीक है, बेनोमाइल, and this both can be protect the plant up to the 90 days. So the management जो है like this are some of the effective control measures that are recommended for the management of the red rot pathogen of the sugarcane. And again we could see the cultural management of this particular pathogen is more important than management with the chemical and the other sources. So this is the chemical, cultural and some resistant varieties related to the control for the red rot disease. Now next pathogen is the smut, that is Stilago scitamania. It is caused the smut disease in the sugarcane. The disease is often manifested by the smutted or like whip-like appearance of the top tissues. So the stem become whip-like and it is fully covered by the smut spores. Okay. So this smut जो spores रहते हैं, the smut spores they can also be seen by opening the unfolded leaves of the stem. So the pathogen, they survive in the infected host. So management practices include cultural use of the resistant varieties and chemical methods. First, we see the cultural method. They include the removing of the whip with the thick cloth without allowing the spores to set any destroy. So once we take out the whip, it should be covered with a cloth, so that the smut spores, they are not distribute or does not fall on the other healthy plants. So this can should be taken, or this care should be taken. The use of some disease free sets are also recommended for the planting purpose, or you can treat the sets with the hot water. It is again advisable that it is recommended for the planting. ठीक है, it is about 52 degrees Celsius for 30 minutes, and we should not take ratoon crop for the infected areas to the fresh crop, because ratoon को इसलिए नहीं लेना है because the ratoon crops may harbor the pathogen and they may be pass on the pathogen to the fresh crop. So resistant varieties are the reported in many areas. So resistant cultivar should be used for the management of this pathogen. Chemical include carbendazole, ठीक, which is the one of the best chemical for the management of this particular pathogen. So आपके एग्जाम में आए तो आप इस तरह से लिख सकते हैं. Now next is the grassy shoot of the sugarcane, and it is caused by the virus. So the pathogen is in the survived in the infected host. This disease is very easy to diagnose where the sugarcane shed does not develop into normal shoot, rather there is a grass like appearance. So this is a very easy to identify. The shoot become grass like in a nature. So it is the only the cultural practices that are help or removing the particular disease. So this include uproot and destruction of the affected clumps, because in the way we avoid the spreading of the virus from the infected plant to the other healthy plants. So we should also avoid taking sugarcane crop for next 3 to 4 year. So crop rotation with a non-host for 3 to 4 year done after 3 to 4 year 3 to 4 year, they has proved very effective in the management of this particular virus. So fresh sowing done after 3 to 4 year with some seed sets from the resistant varieties, and some resistant varieties are reported in for the control of grassy shoot. So after the 3 to 4 year of the crop rotation, one should go for the utilization of the resistant variety for taking a fresh crop. And like we can also do set treatment with hot air at the hot water 54 degrees Celsius like for 8 hours to inactive the causal agent virus is again recommended, as it is the another easy way of managing this particular disease. And next is use of some disease free sets for the planting is again important. Hot water treatment is also effective, where we should treat the sets at 50 degrees Celsius for at least 2 hours, and control of aphid by spraying suitable insecticide, it is also recommended, because the virus is aphid transmitted. तो ये याद रखना, this virus is transmitted by the aphid. So we can avoid the spreading of the virus through effective management of the aphid. Now last one is the red stripe, and it is caused by the bacterium that is Xanthomonas albineus. So red stripe, it is easy to diagnose because here is the developed on the leaf instead of a single red middle portion. So here you can see that the different strips are developed. That's why called red strip. ठीक. दिस स्ट्रिप किस में डेवलप होगा? इन द ऑन द लीफ. So the pathogen survived epiphytes on sugarcane phyllosphere. So this is the main mode of the survival of the pathogens. Now IPM practices include mostly the cultural practices, as there is no recommended like chemical that are available for the management of this particular bacterium. When the affected plants are few in a number, then we can easily take out this affected plants and we can destroy them or even burn them also. But if the whole field is affected, then we should discard the field for at least 3 or 4 years for sugarcane cultivation. Now fresh sowing should be done with the some use of resistant varieties wherever the source is available, and it help in the reducing the disease severity. So this is very difficult disease sugarcane to control. Therefore, whenever disease is noticed, the affected plant should be removed immediately and burned immediately. Otherwise, once the pathogen is established in the field, it is very hard to remove from the field, and that is that is why its establishment should be avoided. Now some systematic destruction of the affected plants, they reduce like the disease incidence, and like growing resistant varieties, they are also the best management practices for the managing this particular disease. So these are the some important sugarcane disease, and this they cause severe loss in the sugarcane crop every year economically. So we should go for the adequate cultural and resistant source for the management of this disease wherever it is applicable, and like thereby we can save the losses that farmers are occurring every year. With this, we have come to the end of the this lecture. So in the next video we will discuss about next topic. So thank